In this video, we're going to look at the function of nucleic acids. That is, DNA is genetic information, and RNA is messenger, translator, synthesis of this information. Now, the main experiment, or one of the key experiments, I should say, that DNA is genetic information comes in 1944 from Avery, McLeod, and McCarthy. Now, there was experimentation before this that was hinting that DNA is genetic information, but scientists were generally skeptic. An experiment that further proved that DNA was genetic information was done using pneumonia. So there's two types of pneumonia: rough strain and smooth strain. So rough strain is non-virulent, doesn't make you sick. So if you take rough strain and inject it into mice, the mice dies. If we take smooth strain, which is the virulent form, you inject it into mice, the mice dies. If you take smooth strain, but you kill all the bacteria with heat, the mice lives. However, if you take this heat-killed smooth strain, mix it with the rough strain, the mouse now dies. And this is suggesting that there's something in this heat-killed smooth strain that heat-killed smooth strain that the rough strain is uptaking and transforming into the smooth strain. And if you use heat, heat denatures proteins, and so it's thought, oh, DNA must be what these bacteria are uptaking to become virulent. So that was in 1944. They're showing DNA is probably genetic information. Now let's look at DNA structurally and think about its replication. So in the last video, we talked about how DNA is a double helix. And this double helix form already proposes a way for DNA to be duplicated. So we know that A always goes with T and G always goes with C. So if you want to create a new DNA strand, a new double helix rather, all you need is one strand of DNA, and you can make a copy of that by knowing which base pairs, which bases base pair together. And DNA copies what's called semi-conservative. That means every new DNA double helix is made out of one original DNA double helix strand and a new strand that is synthesized. And with this base pairing, you allow for accurate replication because let's say, for example, you have an A in this original strand and you try to put a G here. Well, A and G can't base pair, and so your replication mechanism will recognize, oh, there's a bulge here. We're not base pairing. I must have made a mistake, and it'll go and fix it. So DNA replication, for the most part, is accurate. Now, how do we get from DNA to proteins? And this is called the central dogma of biology. So DNA can replicate itself, like we saw in the last slide. DNA can then go through transcription to become RNA. This RNA can be translated to protein. So it goes DNA, transcription, RNA, translation, protein. In DNA, each three bases of DNA can correspond to one amino acid, and these pairs of threes are called codons. So generally, the way that this works is that DNA is transcribed into mRNA. This mRNA is then translated to proteins through tRNA. So each tRNA recognizes the mRNA and carries one amino acid. And this mRNA and tRNA joined together to make a protein happens in the ribosome, thanks to ribosome rRNA. So let's look at this translation a little more closely now. So the mRNA is read into the ribosome. And in the ribosome, tRNAs are naturally floating around, and they come to the ribosome to match up the mRNA. And they have an amino acid with them. And they take their amino acid, and it basically grows onto a chain as shown here. So here's the old tRNA that's ready to leave the ribosome, but before it does, it will pass off this chain to this next amino acid. And so, although the mRNA is what is being read in, this mRNA was copied from the DNA. So, any mutations in DNA will be carried over by mRNA and will show up in your proteins. Because if you have a mutation, your tRNA doesn't know that's supposed to be wrong, and so it'll bring over the amino acid that your mRNA says. And with this idea, we have some vocab words here. So if you study genomics, you're studying a genome size, organization, and the gene content. While if you study transcriptomics, you are studying gene expression. And proteomics is the study of protein content inside of a cell. And generally, a genome of an organism won't change. Whatever DNA you're born with is the DNA you're going to have. While the transcriptome in the proteome will vary greatly based on age and what tissue you are in. So, although the genome itself doesn't change, different regions can be turned on and off, and this will affect the transcriptome and proteome. And that's it for this video. I will see you in the next video.